All right, folks. Welcome to the last session on the last day. They did, in fact, put plant reproductive biology, so save the best for last. My name is Bridget Bickner, and I am a PhD student at Harvard University. I work with Robin Hopkins and Elena Kramer. And broadly, I study the evolution of seed packaging variation in phlox. So for any folks in the audience who may not be plant biologists, these are probably some more familiar fruits in your vocabulary. So on the left side of the screen, you have an avocado. On the right side of the screen, you have a papaya. And a really striking difference between these two horticultural plants is that avocados are making one very large seed, but papayas are making lots of smaller seeds. And this difference in strategy is thought to represent differences in investment in offspring quantity and quality between not only these two species, but any species that have this type of variation. A lot of the variation we see in seed size and number is found at higher taxonomic levels. So this is variation that's found in the legume family. But we very rarely see variation in seed size and number occurring at smaller taxonomic levels, meaning in species or in genera. And most of that is just simply due to similarities and relatedness and developmental components relating to how to produce a seed. So understanding the evolutionary mechanisms that drive the evolution of seed size and number is challenging because of this. And so it's really exciting to be able to work in a system in which we have variation among closely related species and even within a species. So just to give you a crash course on how to understand ovule packaging in phlox, phlox produce 3 chambers per ovary. And so what you're seeing on the top part of this diagram in the circular structure are these 3 different chambers with 1 ovule forming in each of those chambers. On the second lower portion of this diagram, you're seeing an exposed locule, and again, 1 ovule is forming in this chamber. So in light green are our ovules. The white is our locule space. The green on the outer portion is representing the ovary wall, and all of our ovules are forming from this central placenta along the central septum. So about 60 of the 65 species of phlox are single ovulate. However, there has been several independent transitions to packaging multiple ovules per locule, and this is what this looks like. There is a mini ovulate phenotype that I see in Phlox romeriana, but there's been several independent transitions to this fewer ovulate phenotype that I call a biovulate phenotype across several other species of phlox. But what you might not have noticed so far is that I've actually listed Phlox longifolia under 2 of these categories, the ancestral state being single ovulate, and this multi-ovulate state being the derived phenotype. And if we think about what's happening in these 2 different strategies, just comparing the single ovulate state to the multi-ovulate state, these multi-ovulate individuals aren't just necessarily producing 2 ovules per chamber. Sometimes they produce 1, 2, 3, or 4, while these single ovulate ancestral state species or populations are always producing 1, and there's no variation in this trait. And so the evolution of this single ovulate morphology to this multi-ovulate morphology is inherently the evolution of a plastic phenotype. And what I'm really excited about with this transition is that I've actually identified a species that is polymorphic for the ancestral state and the derived state. And this work started all from 2 sentences written by Asa Gray in 1870 describing this variation in ovule packaging between what he was terming at the time 2 different species. This taxonomy has since been redescribed, and it's no longer considered to be 2 different species, but he was originally describing that some plants always have 2 seeds per chamber, while other plants are always producing 1. So I started this project by going to the herbaria, and I sampled from a bunch of different specimens from across the species range, including some of the same specimens that Asa Gray worked on all the way back in the late 1800s. And what you might notice from this central picture of a dissected fruit is that there's a ridge along that dark brown part, and that ridge is a separation between these 2 different seeds. So that's actually 2 seeds in a chamber on that particular fruit. And so from this herbaria work, I identified, or I described a broad distribution pattern within this species, identifying populations that appear to be single ovulate and populations that appear to be biovulate, as a way of preparing myself to go into the field and try to find these 2 different morphs. So I did do some exploratory field work, and I was able to show for the first time in the field, looking at live plants, that there is variation in ovule packaging within this species, where there are some populations that have a single ovulate morph, which is shown on your left, and other populations that have this multi-ovulate morph. Again, from some of this work, I was able to show that there is no difference in fruit size between the 2 different morphs, and that the variation that we're seeing between these 2 different morphs is that seed size is what's variable. And so this very clearly shows the trade-off between seed size and number between these 2 different morphologies. And so the question that I want to ask in this particular project is, how does environmental variation produce and maintain diverse reproductive strategies? And there's a variety of different ways that we might expect this could happen, especially in a species with a relatively wide spatial range. Some variation that might be relevant to plants is moisture, temperature, UV radiation, or differences in the growing season start, end, or duration. We also might expect that pollinator service differs across space, or even pollinator identity might differ across space as well. And so in this chapter, I'm breaking this down into these 4 other smaller components or chunks. First of all, asking what is the spatial distribution of the morph frequency across space? How does environmental variation play a role in this particular polymorphism? Is there a correlation with other floral traits that might be important for understanding this transition? And also, what is the role of plasticity, particularly where we get from this ancestral state of a fixed phenotype to this new plastic multi-ovulate state? But we'll go ahead and start with just B and C before we get into A and D. So as part of my fieldwork, I sampled from 108 populations across 8 states. This represents most of the species range, with the exception of the Sierra Nevadas. And I sampled from 13 of the commonly recognized 15 subspecies in this particular species or species complex. I also was able to sample from populations with variable elevation, anywhere from 200 meters above sea level to 2,700 meters. I sampled from populations that were flowering as early as April and other populations that didn't start flowering until July. I sampled from diverse ecological communities. Most often, this species is found associated with sagebrush communities, but not always, so this is an example of a population found in a woodland environment. And I also took note of all of the different abiotic variables that might be responsible for determining variation in spatial distribution. Some of these populations are really variable in floral morphology as well. So this is a particular population with corolla tubes that are more than 2 inches in length, which could very easily exclude some particular pollinators. And also taking note of any pollinator activity that I see at some of these populations. Okay. Before we get into the actual distribution of this phenotype, I want to take a quick dive into some of the other morphological traits that might be relevant here. So the presence or absence of glands on the sepals has been used many times to describe these different subspecies and is considered to be a key break in how to determine the identity of these different subspecies. So I first wanted to ask if there was variation across these different sepal morphologies for these 2 different morphs. So what we have on the top is a glandular pubescent morphology, so these are hairy and sticky. In the middle, we have glandless pubescence, this is hairy and not sticky, and on the bottom, we have no pubescence or glands. So I wanted to ask, are multi-ovulate plants found in some subspecies and not others? And the answer is no. We find both morphologies present in all 3 of these subspecies categories. Okay, let's get into the distribution patterns that we're seeing with these 2 different morphs across the species range. So this is what I observed, and what we're showing here is just presence or absence of the multi-ovulate phenotype at each of these sites. We'll do a deeper dive in just a minute, though. What I want you to see, or at least what I've observed in this particular data set, is that there appears to be these two different clusters, one in the north and one in the south, that I'm calling the northern group and the southern group. And there doesn't appear to be any multi-ovulate populations in the central part of this range, which is really curious. And so some of the questions I have surrounding this that I unfortunately don't yet have answers for are, why aren't there multi-ovulate populations in the middle of the range? And what do the northern group and the southern group have in common? If we take a closer look at a subset of these populations in the northern group and the southern group, we also see that the frequency of the multi-ovulate phenotype is really different in the northern group and the southern group, where a lot of our multi-ovulate populations in the north are very strongly multi-ovulate. Almost every individual in the population is displaying this phenotype. And the variation that we see between the multi-ovulate and the single ovulate population could be due to genetic variation, but it also could be due to plasticity. In the southern group, we're much more often seeing the single ovulate phenotype, even among populations that have the multi-ovulate phenotype. So are these more frequency differences between the multi-ovulate populations due to genetic variation, plasticity, or both? And that's a question that I have yet to answer. What's going on in the southern group? We have this mixture of multi-ovulate and single ovulate populations occurring in really close proximity to each other. So I just wanted to see what sort of variation might be responsible for that. So we don't see any difference in elevation between populations that are fixed for the single ovulate and populations that have the multi-ovulate phenotype. We do see a relationship between elevation and flower size, so flower size increases with elevation. We don't see a relationship between flower diameter and seed packaging, but it does appear that the multi-ovulate plants do have larger flowers and less often smaller flowers. And so to just wrap up, multi-ovulate morph is found in the northern part of the range most often, and among some populations in the south, the variation is mostly between populations, but some populations may be mixed. There's notable variation in the abiotic conditions across these different sites. Ovule packaging variation does not segregate with subspecies identity. There's no difference in flower size between these morphs. However, the multi-ovulate populations rarely have small flowers. And we don't yet have an understanding of the role of plasticity in this species, but I'm really excited about some continuing directions in this species. So I'm currently growing up 3 different population pairs in the greenhouse, specifically to look at does the source population predict the morphology that we see in the greenhouse? Are we able to produce viable offspring when crossing between these 2 different morphs? And how is plasticity operating in this multi-ovulate phenotype? And so with that, I would really like to thank my dissertation advisory committee, the Kramer Lab, for their helpful feedback on this presentation, the Harvard University greenhouse staff, all of the support that I had with permitting for access to the land for these field collections. And I'd like to give a special shout out to Eric Rolson, Simone Pesce, and Dan Schoenberg for their help in the field. Thank you so much.
Hi, everybody. My name is Grace Burgin, and I am a graduate student with Dr. Robin Hopkins at Harvard University. And I am very happy to be here today to tell you a little bit about some of my dissertation work investigating the genetic basis of self-pollen recognition in the wildflower Phlox germundii. So flowering plants occur along a distribution of selfing rates from near complete selfing to obligate outcrossing. And where a species falls on this distribution is really important because selfing rate determines how genetic variation is distributed across both space and time, therefore affecting the evolution of all other traits across the entire genome. And while there are many adaptations or traits that affect a species' selfing rate, some of the most common occur post-pollination inside of the flower and are mediated by interactions between the reproductive organs, so specifically pollen, which carries sperm, and the pistil, which houses ovules. And in cases where self-pollen, so pollen from the same individual, arrives at the pistil, over half of flowering plants have genetic mechanisms to specifically reject self-pollen thereby preventing self-fertilization and limiting the costs of inbreeding. But in order for self-incompatibility to function, a pistil must be able to reliably recognize self from non-self pollen. So how does this work? Well, it works through the action of something called the S locus, which is a single genomic region that contains both pollen and pistil-expressed genes, which interact biochemically at the pollen-pistil interface, such that if pollen and pistil have the same S locus allele, the cross is recognized as self, it's incompatible, and no fruit develops. Whereas if pollen and pistil have different S locus alleles, the cross is recognized as non-self, it's compatible, and fruits will develop. And while this general pattern for how S loci work is shared, it's actually evolved independently, likely 14 times throughout flowering plant evolution. And at present, we know the genetic identity of the genes within these S loci in just 4 of these evolutions. And we know them primarily from model organisms. And what's really striking about this is that both the genetic identity and the molecular mechanisms of these S loci are super, super different across these different independent evolutions. So if we really want to understand the diverse mechanisms through which flowering plants achieve this self-discriminating phenotype, we need to look more broadly across the angiosperm phylogeny. So this is what I do in the wildflower Phlox stromundii, which is in the family Polemoniaceae. Based on its phylogenetic position, you have to forgive my highly pruned phylogeny. It's to make a point. And based on its position, we initially hypothesized that Phlox may be using an sRNase-based SI mechanism. So to test this, I generated RNA-seq data from 5 pistils, screened it for expression of sRNases, and found absolutely no evidence of expression. Moreover, when I look for expression of these other known S loci genes, I find, again, no evidence of expression. And so what this is telling us is that there is a currently uncharacterized S locus that has evolved in the lineage containing Phlox. So how are we going to find it? The approach that we took is to start by using genetic mapping to identify the genomic region containing the S locus. So to do this, we tricked a single individual of Phlox into selfing a whole bunch of times to generate 98 selfed offspring, which recapitulate these 3 possible genotype crosses. We can then cross them all to each other to infer what their S locus genotype must be. I then took a bulk segregant association mapping approach. So here I'm showing you the 7 chromosomes in Phlox, and on the y-axis is this G' statistic, which associates regions of the genome, in this case with S locus genotype. And what I find is that a single genomic region significantly associates in this mapping population. And when I repeat this with a second independent mapping population, I again find the same region associates with S locus genotype. This is great. And it tells us pretty confidently that the gene causing self-pollen recognition is somewhere in this genomic region. The downside of this is that there are 633 genes that annotate to this region, and so my next challenge was to figure out how to go from those 633 genes and filter it down to our 1 pistil-expressed gene responsible for recognizing self-pollen. So to do this, my strategy was to develop predictions based on what we know about how other S loci function and evolve. So my first prediction is that this gene must be highly expressed in the pistil and not really expressed anywhere else in the plant. So to test this, I generated transcriptome data from 5 tissue types from throughout the plant. Here I'm showing you expression data for the top 15 most highly expressed genes within that bulk segregant region. And when I look at expression patterns across the plant, I see that just 2 of these genes are both highly expressed in the pistil and restricted in their expression. Okay, so that helps us filter, but we want more. My second prediction is that I'm looking for a gene that is highly polymorphic, and this is because S alleles evolve under negative frequency dependent selection. So if you're a rare allele, you have more potential mating partners, and so over time, lots of polymorphism is maintained in the population. And one of the classic ways to screen for polymorphism is to construct gene trees where genes that have a short overall branch length have overall low polymorphism, whereas those with long branches indicate high polymorphism. So I'm really looking for a gene that has a super long overall total branch length. So I assembled gene trees for all of the genes in this region, and I find that there is one gene that is 10 times more polymorphic than all of the others in this peak, and it happens to be one of our highly and specifically expressed genes. Okay, so we have a candidate that matches these two predictions, but of course, one of the most important things is that genotypic variation at this gene must predict cross-compatibility when you cross individuals to each other. So to test this, I worked with a really phenomenal undergraduate collaborator, Nia Lewis. We took 2 individuals and crossed them to each other to generate 10 offspring with 4 possible S-locus genotypes. We then crossed them all to each other in a full diallele design. And by doing this, we recovered 4 crossing groups consistent with these 4 possible genotypes. We then designed allele-specific primers to genotype these individuals, and what we find is that genotypic variation at this gene perfectly predicts cross-compatibility in this full diallele cross. Okay, so collectively, this is a great candidate, I think, but of course the ultimate test is functional validation. So in other words, if we knock gene 52568 down, can we take a self-incompatible plant and make it self-compatible? So my approach to doing this was using virus-induced gene silencing, or VIGs. And one feature of VIGs is that it doesn't cause silencing on every flower of a treated plant. And so I needed some way to know whether a flower was actually having silencing. So my strategy was to use a floral color gene, so construct a vector that has a target for both my gene of interest as well as a floral color gene. And by doing this, I can compare within each individual white flowers, which are silenced, compared to pigmented flowers, which are unsilenced. Okay, so first I will show you the data from my control experiment. So in this case, I treated plants with just this floral color gene. And then when I self-pollinate those flowers and compare the white versus the pigmented flowers, I see absolutely no difference in the proportion of self-fruits that set. Then when I take S4 homozygous plants, so this line of S4 homozygote plants, and target this allele S4 specifically, I find a significant increase in self-fruit sets following self-pollination. So this is really exciting because it tells us that by silencing gene 52568 alone, we can take a self-incompatible plant and make it set self-fruit. In a related but slightly different experiment, this time I treated heterozygous plants, so S2, S4 heterozygotes, but again targeting S4 specifically. So in this case, only S4 is silenced while S2 should still be active. I then pollinated these plants with pollen of known S-locus genotypes. And what I find is when I apply S4 homozygote pollen to these plants, we see an increase in compatibility. In contrast, when I apply S2 homozygous pollen to these plants, we see absolutely no increase in compatibility. And so what this tells us is that this gene has allele-specific activity, which tells us that it is involved in self-pollen recognition. Okay, so I'm hoping at this point you're all really desperate to know what gene 52568 is. It is a G-type lectin receptor tyrosine kinase with a single pass transmembrane domain. And initially, when I saw this, I was really surprised because this is a member of the same gene family as SRK, which is the gene causing self-pollen recognition in a super distantly related family, the Brassicaceae. And so I think that while at present it may appear that the genetic identity for how flowering plants achieve self-pollen recognition is really diverse and disparate, as we continue to identify more of these S loci, we may be able to learn from commonalities or repeated patterns to better understand how self-incompatibility evolves and functions. So to end today, I would like to zoom out a little bit because what I really love about studying self-incompatibility is that it has the potential to teach us about a lot of really fundamental biological processes. So this work is just a first step towards better understanding the genetic basis and mechanisms of mating system variation, to learn about how genes with interesting function evolve and where they come from, and then also to understand how complex cellular communication genes either evolve together or separately. So I'll thank the research community that supports both me as a student and this project, particularly acknowledging my co-authors, Nia Lewis and Dr. Andrea Berardi. I want to thank you all for being here this late in the conference, and I'm happy to take your questions.
Thanks everybody for coming. My name is Jeremiah Busch, and today I'm going to give a talk where the basic idea we're going to try to figure out is, why do organisms go through all the effort to find another mate instead of just mating with themselves? And this is a problem I've probably thought about too long. But I also think that plants are wonderful systems to work on because they are hermaphroditic, and so they're actually tasked with that question: should I self or should I outcross? So before I get started, I just want to point out Shelby here. This work I'm presenting is her first dissertation chapter, and so it's very much a collaborative work, and she deserves much of the credit here. And stay tuned, there will be lots of great work coming from her after this. So just to introduce this problem, so Stebbins, I'm putting up a couple quotes here from him, noted a few patterns wherein reproductive systems seem to evolve in very particular ways. And the first quote is essentially that selfing evolves frequently from outcrossing. And in fact, it's one of the most common transitions we see. And so one way of making sense of that is to compare these different organisms shown here. This is work by Dan Schoen, where you have large flowered individuals on the left, which have distyly, and small flowered individuals on the right, which self-fertilize primarily. And what you often see is that the selfing lineages are at the tips of these trees, so they've been derived recently. And the question then becomes, well, why is that the case? And Stebbins made the case later, this lower quote, that perhaps there's a genetic problem to this close inbreeding over time. And the essential logic is those gene pools are really shallow, and because there's not a lot of heterozygotes, there should be infrequent recombination. So maybe these lineages, once they adopt selfing, just lack the capacity to adapt to changes that arise. And that's what might drive them extinct. And so in this talk today, there's something else buried in here, and that is once you evolve selfing, you don't go back to outcrossing. That's a big component of this hypothesis. And so I want to test here, or we want to test here, whether or not selfers lack the genetic capacity to re-evolve outcrossing morphologies. And so that is what I just said. So that's the question for today. To what degree is that transition really irreversible? And so just to convince you with some more compelling examples, I'm showing you two pairs of close relatives. On the left, these are Capsella species, where the species on the left is self-incompatible, large-flowered, needs a mate, okay, has to find a mate. And Capsella rubella on the right does not have this requirement. Selfs readily, has really reduced petal sizes. We see these transitions even within species in these two Clarkia. So on the top in these black and white photos, we have a large flower, note the scale, and there's a big separation in space between the stigma and the anthers. So it's more difficult for self-fertilization to happen there. Whereas the subspecies on the bottom has those two characteristics in really close proximity, making selfing really, really capable or easy. And so I think when we ask this question, are these transitions to selfing irreversible, I think we can all pretty much accept that self-incompatibility systems, given their complexity, are rarely regained. But in all cases, for both of these pairs, what we have is adaptations for self-fertilization that are probably of a quantitative genetic basis. I mean, this is the stuff of mating system evolution, and so it's going to fuel transitions to selfing and back. And so essentially what we want to do is ask, can we make selfers look more like outcrossers, or do they lack the genetic capacity? So to test this idea, I'm going to work in the yellow monkey flower species complex, Mimulus guttatus. So in this map in gray, you can see its very, very large geographic range. There's been a lot of work done in this group. And in monkey flowers, there is tremendous variation in mating system, or the degree to which plants self-fertilize. So Mimulus guttatus, or Erythranthe guttata, tends to have an outcrossing / mixed mating strategy. There's lots of variance there. I'm reporting a couple of the selfing rates from long ago. And within this group, there's a population known as Joy Road, which Kermit Ritland worked on a long time ago. And it was very interesting to him because it tended to have a very high selfing rate and had a curious stigma-anther distance. And so the work here, if we step back and ask, well, how is this related to maybe all the diversity in this genus? This Joy Road project is really just a first step towards maybe doing this more broadly. Okay, so one thing you might ask is there genetic variance in general for floral characters in E. guttata? The answer is a resounding yes. Maybe there's a publication effect here, but every selection experiment that's out there produces sizable responses from many different groups. And so there's a good historical work there that we can compare this Joy Road population to. Okay, so just to orient you on exactly what we're doing here, so on the right is a monkey flower that's opened up, and so you can see the organs. And so what we're going to be really focusing on here is that distance between the stigma and the anther, because that's the functionally really important characteristic in terms of influencing the selfing rate in these lineages. And so if you measure that distance in two different populations, Joy Road and another Guttatus from Yellowstone National Park in the same greenhouse, you'll see the histograms on the left. So the Yellowstone plants have the stigma above the anthers, okay? This makes selfing unlikely, whereas the Joy Road populations have the opposite orientation. The anthers are above the stigma, causing self-fertilization readily. Okay, so we did whole genome sequencing on a few of the Joy Road individuals just to make sure that they are Guttatus. Of course, that question in and of itself is very complicated, but they are very close. Their closest relatives are other Mimulus guttatus from the southern clade nearby on the Pacific coast in Oregon and California. So we think it's natural to do this experiment and then compare our results to other guttatus work. Okay, so what did we do? Simply put, we used the selection experiment as a way to extract a component of the genetic variation. That's the whole purpose, to figure out if selfing reduces it. And so what we did is took 50 mothers grown from seed and made a very genetically variable source population called G1. That seed was then split into 4 different populations, 2 controls in the boring colors, 2 selection populations in the warm colors. And so we subject the selection populations to 5 generations of positive selection on stigma anther distance. And then at the end, the G7, we compared those evolved descendant lines to the ancestor. And so just some more details here. Every generation, there were 200 plants per population, and we always chose 50 plants to be parents. To make those decisions, we measured a bunch of floral traits, and they didn't inform the decision in the control lines, but they did inform the decision in the S1 and S2 lines. And that's basically shown on the right. We took the upper 25% of the distribution, and they then were used to make the next generation. Okay. So what did we find? A lot of work in one slide. And so, again, the boring lines are control, that's gray and black, and the warm lines are the selection lines. We see not much separation between the control and selection lines until generations 5 and 6, where that becomes significant. One thing you might naturally ask is what's going on with generation 2? That's what I always think when I see this slide. This was a pandemic project where we had trouble getting lights in our greenhouse that time, so the plants turned into mini bushes. Nevertheless, we produced a selection response near the end of the experiment, but if you look at this and think back to other selection experiments you might have seen in more outcrossing Mimulus, you will note that the separation is very, very small. So if you raise down those descendants, the G7s, the evolved lines, C1, C2, S1, and S2, and compare them directly to their ancestor in a common garden, we can then get at the response. So the way we define the response here, because they're all growing in the same environment, is to take the descendant mean. So for instance, look at S2, that yellow distribution, and the mean there, and we subtract off the ancestor. That's the response to selection in that population. And then we can just basically rearrange the breeder's equation to get the realized heritability. To do that, we take that response and divide by the cumulative amount of selection we put on that population. So after doing that, if you look at the realized heritabilities in both the S1 and the S2 lines, you'll note that they're very small. So those are realized heritabilities below 0.05. And if you look at the standard errors, it's actually very hard to say they're not 0. So that's pretty small. And in terms of the comparison, it's not perfect, but Holesky and Kelly did a very similar experiment, which we emulated, and they had an index of flower size and flowering time, and they found realized heritabilities that were much higher, so 0.18 to 0.33. So that's about an 84% decline in realized heritability. Okay, so we also use our data just to kind of compare a few models of phenotypic evolution. One of these you might expect is let all the lines be defined by some common mean phenotype, the stigma anther distance, and that, yeah, there might be drift, but they're all defined by that same mean. And we can compute the probability of the data by just looking at the individual data points and assume a normal distribution and calculate those probabilities. So you can do that for this model here, which is genetic drift. You can do it for a more complicated model here, which you might call response to selection, where the S1 and the S2 populations are defined by a different mean of a normal distribution. You can do this for a whole universe of ideas. Well, not a whole universe, because there's not that many populations. But you could do it for others, such as adaptation to greenhouse, where all the descendant lines or populations are the same and the ancestors different. You could imagine multiple responses to selection where S1 and S2 differ, or idiosyncratic adaptation. And if you compute the probabilities, what you find is that the simple response to selection model is the best fit, though idiosyncratic adaptation is a very close second. Okay, so what does this all mean, and what's the advance here, if anything? When you look at other works where people have compared outcrossers and selfers, in fact, this has been done in Mimulus amongst very close relatives. So Carr and Fenster did this comparing Mimulus guttatus and the highly selfing Mimulus micranthus, and found large reductions in total genetic variance, including stigma anthodistance. So this is supporting that finding. Clow et al. Summarized studies across angiosperms that looked at genetic variance, particularly the heritability, in lineages, and found also similarly largest reductions in that component of genetic variation. So one thing you might think is, well, maybe stigma anthodistance, in your study, you have an 84% decline in the realized heritability. Maybe that's because this trait has a history of selection on it. And so Opedal in 2017 actually looked specifically at the heritability of hercogamy, which is essentially stigma anthodistance, putting together all the studies that have been done. And what they found is actually not a tremendous loss of variation in this trait. There's still a fair amount of variation in hercogamy, even though you might think it would be a target of selection and therefore have less genetic variation. Clearly, our work adds to this, but there's a lot more to do. In terms of the bigger picture, in terms of remembering the question, the whole idea was, once selfers commit to this reproductive strategy, can they go back? And you might ask, well, what does our experiment actually mean? We did a greenhouse experiment where you subtly change the stigma anthodistance. Is that actually going to influence the mating system? I would say that's not obvious and it probably won't. So those connections need to be thought a little bit more about. And like many talks here, I think we still need to work on connecting these observations we make in controlled settings to differential survival and extinction, say, in nature. Because that's really what that hypothesis is all about. Committing to this close inbreeding leads to the loss of lineages. I would also say that this whole talk has been an obsession about genetic variation. I think most talks tend to be in evolutionary biology. But I would also say that maybe selection is less powerful in highly selfing lineages too. So selfers, if they don't have inbreeding depression, there is no selection to actually outcross. So that's another thing to keep in mind. And also in highly selfing lineages, if there are no pollinators or opportunities for outcrossing just don't exist, there needs to be individual variation for selection to operate. And so that hypothesis is worth looking into. Okay. With that, I'd like to thank everybody, and especially Shelby and collaborators on Monkey Flowers, and you for spending the time here. Thanks.